My name is Savannah Hess, and today I'll be presenting a paper written by me et al. Titled A Role for Bacterial Urease in Gut Dysbiosis and Crohn's Disease. This paper explores the effects the microbiota has on Crohn's disease, specifically the role of bacterial urease and how it plays in the development and continuation of dysbiosis. First, I'll go over the basics of what Crohn's disease is before we dive into the paper. Crohn's disease is an autoimmune disorder that causes inflammatory bowel disease. It is characterized by chronic inflammation in both the small and large intestine. This inflammation causes a host of symptoms that vary between patients, but often include weight loss, fatigue, abdominal pain, and diarrhea. The exact mechanism by which Crohn's disease occurs is still unknown, but it is thought to occur due to an interaction between genes, as a genetic predisposition is required to develop the disease, environmental factors, such as diet, antibiotics, stress, or smoking, the immune system, and the microbiome, particularly dysbiosis versus homeostasis. Although homeostasis is a relative term in this case, and a healthy composition for one person will be very different from the next. The rates of IBD diagnosis have been steadily increasing each year by almost 6%, and has skyrocketed by over 50% between 2000 and 2010. Most individuals with IBD are diagnosed before their 30s, but this age of diagnosis is dropping as childhood occurrence rates are increasing. Current treatments only manage symptoms to improve the patient's quality of life and allow for nutrient absorption. Treatments can include monoclonal antibody injections that dampen the immune system, and these are often paired with lifestyle changes such as diet and exercise plans. But these medications often come with a host of side effects ranging from fatigue and even cancer development. New research is being done to examine the effects of fecal microbial transplants to reverse dysbiosis and reduce symptoms of IBD. Many studies, such as that conducted by Lewis et al. In 2015, have shown that IBD patients often exhibit dysbiosis that may be caused by and contribute to intestinal inflammation. This is associated with a general decrease in the richness of the microbiome, including bacteria from the phylum Firmicutes. Firmicutes are required for the production of short-chain fatty acids that support immune tolerance to the gut mucosa, thus acting in a protective manner against IBD. This decreased alpha diversity is often paired with increased abundance of taxa in the proteobacteria phylum that has been associated with gut inflammation in animal models of IBD, as reported by me et al. With an unknown cause and rising cases, IBD is becoming a major burden on our healthcare system, not to mention the burden that this disease has on the affected individuals each and every day. Patients often need both medical and surgical interventions if their disease is not managed properly. Because of this, each year Canadians with Crohn's disease cost the healthcare system about $1.28 billion. Biologics are extremely expensive, and Canadian citizens often rely on government funding to afford their medication. And this medication, again, does not offer a cure, but rather just temporary relief from the symptoms. In this study by me et al., they aim to find a connection between fecal amino acids and dysbiosis in IBD patients. They are specifically examining nitrogen metabolism from bacterial urease. Bacterial urease is a factor found on bacteria that have the capability to infect a host. And as the name suggests, it has an enzymatic function that allows it to damage human tissue. The authors hypothesized that bacterial urease may affect the gut microbiome after proper cleansing with antibiotics and polyethylene glycol, or PEG, to reduce biomass in the gut. Therefore, this study is very important for the world of gastroenterology. If a mechanism can be established between bacterial urease and IBD, then possible treatments can be the next course of action. Me et al. Drew data from a previous study called PLEASE, outlined in the paper by Lee et al. This study used metabolomics and shotgun metagenomic sequencing on samples from participants under the age of 22 with active Crohn's disease. In this study, stool samples were obtained at baseline and then 1, 4, and 8 weeks after starting their treatment. Treatments include partial enteral nutrition, PEN, exclusive enteral nutrition, EEN, or anti-tumor necrosis factor alpha. Me et al. Used biospecimens from this study to relate their mouse models to humans. Me et al. Performed a multitude of methods to identify a relationship between fecal amino acids and IBD progression. They used a combination of heavy isotope nitrogen flux studies, bacterial culture experiments, and mouse models in their studies. Urea with nitrogen-15 was incorporated into bacterial amino acids. Then E. coli was engineered to express urease and introduced to mice that were treated with antibiotics and PEG to determine the effects on the microbiome. Later, RAG2 knockout mice were inoculated to determine the functional effects on IBD development. A derivative of murine commensal E. coli, MP1, was engineered to express an inducible urease operon, and when its activity was confirmed, it was placed on the mice 5 days after cleansing with antibiotics and PEG. This gut cleansing allowed for mice to be successfully inoculated with the engineered murine E. coli, referred to as URE+, or the wild-type murine E. coli, which is referred to as URE-, or a 1-to-1 ratio of URE+ and URE-. Antibiotics PEG and antibiotics PEG colonized with species from conventionally raised mice were used as a control group. At baseline, the mice had similar compositions of microbiota, as shown with 16S RNA sequencing, but the most significant results were recorded after 29 days, where the URE+ and the URE+- mice diverged from the other groups with large effect sizes. As shown in Figure 5F, these last two columns that represent the mice with the engineered murine E. coli have increased abundance of proteobacteria when compared to the URE- mice, which have higher abundance of Firmicutes. This is significant as it shows that E. coli can be vital in the establishment of a stable microbiome when introduced into a gut-cleansed host, and the presence of bacterial urease can influence this new gut composition. As mentioned in the previous slide, the URE- mice had increased abundance of Firmicutes in their microbial community. This composition has been associated with a healthy gut in the host. On the other hand, the URE+ or URE+- mice had microbial compositions that are associated with dysbiosis and IBD, including bacteria from the Proteobacteria phylum. To identify the functional effects the recorded dysbiotic microbiota had on colitis development, Ni et al. Inoculated mice with URE- or URE+ in a T-cell adoptive transfer model of murine colitis. After 44 days, the URE+ MP1 mice displayed characteristic signs of IBD. This included decreased colonic length, chronic inflammation in the colon, weight loss, as shown in the top figure here, where the URE+ mice are denoted by the red line, and damaged epithelial lining, as shown in the bottom figure here. Additionally, the researchers noticed increased fecal amino acids when compared to URE- mice, much like what was reported in the PLEA study in humans mentioned previously. These results confirm that inoculation with E. coli engineered to express bacterial urease can lead to the development of colitis in a murine model of IBD. This study highlights the importance that one bacterial strain has. Bacteria that express bacterial urease have the potential to induce and worsen IBD in a properly prepared host. This is a very important finding as antibiotics are prescribed every single day and thus provide the gut cleansing that can allow bacterial strains, like the one mentioned previously, to colonize and lead to disease. As shown in the data derived from the PLEA study, the abundance of proteobacteria is increased in individuals with Crohn's disease, and these bacteria are enriched with genes that promote the utilization of nitrogen. This supports a hypothesis that bacterial nitrogen metabolism plays a role in the development of IBD. This study provided crucial connections between the bacteria in the gut and Crohn's disease. However, they did not consider the effects that other members of the gut would have. Fungi that are present in the gut also play important metabolic roles, including nitrogen metabolism, that will play a role in the development of IBD through this pathway. Additionally, they only use E. coli as a bacteria of interest. Other bacteria should be studied to determine if the same results can be observed. Lastly, the study was not able to identify the mechanism by which bacterial urease causes dysbiosis in mice or humans, nor did it determine if all the cases of Crohn's disease are due to this urease-induced dysbiosis. The next steps following this study should be to determine the exact mechanism by which bacterial urease is able to induce Crohn's disease. As bacterial urease is an enzyme, it is possible that it's cleaving a molecule in the gut and thus making it toxic to the gut epithelium. Damage to the epithelium would allow bacteria from the gut to interact with the immune cells that reside in the tissues, thus launching the characteristic autoimmune response that is associated with Crohn's disease. If a mechanism can be established, then therapies could be developed to target the pathway and potentially reduce the dysbiosis and Crohn's disease symptoms. This slide shows the additional resources that were used to create this project. As mentioned at the bottom of the slide, all images were created with biorender.com unless cited otherwise. Thank you.